All right, so for today's note set, there's not going to be that many notes. It's just mainly going to be me explaining how to do your calculations for today. But there is a short section of notes, so I will still provide the extra credit section where you can turn in your notes. And then let me go over the agenda and the homework. Not all of it's due today, so I'm just warning you beforehand that you will have stuff that is assigned today, but due at the end of the week. So don't just cram it all in at the end of the week, because some of y'all experienced that last grading period and you realize that it doesn't really work, and then your assignment quality isn't as good, so that your grade on it isn't as good. All right, so for our agenda, we're gonna first, the order is a little wrong, but we're gonna first do water potential calculations after we do a little bit of notes, and then we'll do molarity calculations, and then we'll go over the diffusion lab procedure 2 that I'm going to be demoing for you all on Wednesday. And then you have your chapter 5 summary and Learn Smart due on Friday. So get a head start whenever you have free time. You have your cell drawings due on Friday, and basically I'm just gonna ask you to look at page 67 of your textbook. There's figures 4.6 and 4.7, and I want you to just draw them for yourself in your notes with the labels. And I'll let you turn them in for not extra credit, for points. So this should be an easy assignment that can help boost your grade and helps you learn the structure of the cells. The water potential calculations and molarity calculations that I'm going over today are going to be due at the end of today. So make sure you turn those two in today. The learning objective is the same as usual. Learners will be able to explain how cell size and shape affect the overall rate of nutrient intake and the rate of waste elimination. So you'll be able to make claims and predictions about natural phenomena based on scientific theories and models. And the second part is mainly because we're going to be doing 2 demos this week, one on Wednesday and one on Friday. So this unit is pretty lab heavy. Keep in mind that there is a unit test coming up really soon, so it's early in this grading period, which does mean you have time to kind of make up your grade after the test, but I would prefer you, just to do better on the test overall to start off with. So let's talk about the origin of organelles, and this is going to be your short notes section. And this is actually a diagram from your textbook. And so if you ever thought about this before, maybe in your regular bio or pre-AP bio class, you talked about mitochondria and you talked about chloroplasts. And mitochondria are the energy centers in all cells, and chloroplasts only exist in cells of organisms that do photosynthesis, because they're the ones that are green and they reflect green light, they reflect green light and then all the other light is absorbed and the energy is used for photosynthesis. So if you look at here, the original cells that actually first started as life forms on Earth are actually prokaryotic, and we have fossil evidence and DNA evidence of that. And so prokaryotes mean that they don't have a nucleus and they don't have membrane-bound organelles, but we know that eventually we evolved over time and that cells now have membrane-bound organelles like Golgi bodies, endoplasmic reticulum. They have mitochondria and chloroplasts. So how did that happen? So somewhere along the lines, a cell gains a nucleus by the plasma membrane invaginating or kind of enfolding and surrounding the DNA with a double membrane. So now that your DNA is surrounded by a double membrane, it's basically a nucleus. So the nucleus allows specific functions to be assigned. So it frees up cellular resources for other work. So when you have a nucleus that has its specific job, it can also assign jobs to other organelles. And being able to be specified means that you can work more efficiently. So that means now that certain organelles in the cells have their own job and they only do that job, they are way better at that job. It's like asking you to be your babysitter for your younger siblings, to be a good student, to be an athlete, to be student council body, to be in marching band, to also be in a gaming club, and then also to work a job. All of those things together is really hard to do, and maybe you aren't able to do your best at all of them because you're tired or you're busy doing the other things. But if you have one job and that one job is to be a good student, then you can do it because you don't have to focus on anything else. And that's the same for the organelles. And then cells gain an endomembrane system by proliferation of membrane. So the membrane is moving around and cells gain organelles like the endoplasmic reticulum that makes packaging proteins and products of the cell to leave or to move somewhere else in the cell. And then there's also like Golgi bodies that help do the packaging, and they make sure that your membranes are being protected as things are moving in and out. So increased surface area allows higher rates of transport of materials within a cell. And like I said, the endoplasmic reticulum, or ER, and the Golgi bodies can do this packaging a lot faster than the cell used to have to do, and pull and drag things around and create the resources, because now it's all there. It's just sitting there waiting to do its one job. And the more surface area, remember we talked about this even with cell diffusion, the more surface area there is, the faster things happen in cells. At some point, cells gain a mitochondria, and this creates the ability to metabolize or break down sugars in the presence of oxygen. And it enables greater function and success because a lot of energy is produced from metabolism of sugar. And this energy can be used to speed up reactions or produce certain reactions that there wasn't enough energy resourced for before. Cells also eventually gain chloroplasts. So the mitochondria was first to be created in a cell from absorbing a bacterium that could already break down sugars. So as you see around this bacteria, there is the cell membrane. And as the cell membrane envelops this bacteria, it becomes a double membrane because obviously the bacteria has its own membrane as well. And we actually still observe that in mitochondria. We still observe a double membrane on the outside of it. So that's why this endosymbiotic theory exists, because there's evidence for it. And so after some cells have mitochondria, some of them also gain chloroplasts from a bacterium that can do photosynthesis. So photosynthesis didn't just magically appear inside of cells one day. It was kind of used from another organism that used to live individually, but now it's in a symbiotic kind of relationship with cells, and eventually it becomes just a regular part of cells that are passed down. And the other reason why we can prove this is because mitochondria has its own DNA. Okay, so the ability to produce sugars from sunlight enables greater function and success. So if you can produce your own sugars that you need to use, because everybody has to use sugars to have energy, and if you can produce your own, that makes you an efficient life form because you don't need to retrieve it from somewhere else. So the more specific reason why we have this endosymbiotic theory is because mitochondria are similar to purple aerobic bacteria. So they both use oxygen in the production of ATP, which is our energy molecule. And they both do this using the Krebs cycle and oxidative phosphorylation. And we'll talk about these processes actually in more detail in our actual cellular respiration and photosynthesis unit. But if you compare purple bacteria and mitochondria, their structures are really similar, their jobs are really similar, their processes are really similar, which is why we think that at some point cells envelop them and now they're just incorporated as part of a cell. Chloroplasts as well. So chloroplasts are similar to photosynthetic bacteria. Both have similar chlorophyll, like the color pigment that does the energy absorbing, that harness light energy to convert into chemical energy. So here's a picture of a cyanobacteria, or a bacteria that does photosynthesis. And here is a picture of our chloroplasts that we see in our plants. See, if you look at them, they're also very similar. So that's a little sus that we didn't just magically develop it, right? Plants actually incorporated these organisms, and eventually they just became a part of the cell. So eukaryotic cells are compartmentalized into organelles. And the advantages of this is because are that isolated reactions from cells are protective. So some reactions are breaking down things and some reactions are building up things. The problem when you're breaking down things is that you're using enzymes that can break down multiple things usually. And if those are exposed to, if these enzymes are exposed to other materials in your cell, then they'll be also breaking those things down. So when you can put them in a compartment or in a specific container within the cell, then they're only breaking down the things in that container. And that makes it a lot safer to do those degradation reactions. Also, the efficiency is higher because you can concentrate all the things that you need broken down inside of that container and then just send in the enzymes and it'll do its job all at once instead of having to have the enzyme spread across the cell and hopefully encounter and come in contact with something it needs to break down. So increased surface area also for more membranes means that you can do more activities. I talked about this earlier with the endoplasmic reticulum, the ER, right? When you're packaging stuff to send out, you don't want to be doing it one by one and slowly creating and pulling the resources. If you have a lot of surface area, you have a lot of surface area where it can be on and doing that job, and then also just creating the membrane and packaging it and leaving right away because it's already pre-prepared. So like we've been discussing, there are two classes of organelles. So there are energy-related organelles like mitochondria and chloroplasts, and then there are the other organelles that are originally just part of a cell that bacteria have, that archaea have, right? So endosymbiotic theory is because mitochondria and chloroplasts have similar size to bacteria. They have circular DNA like bacteria. They have ribosomes, a double membrane from being enveloped by a eukaryotic cell. Because one of the membranes is part eukaryotic and the other membrane is bacterial membrane, right? From the outside originally that came on the bacteria that used to be chloroplasts and used to be mitochondria. And they do their own splitting or fission reactions, right? They have their own DNA and they do their own splitting reactions. So it's like they're literally their own independent organisms living inside of our and plant cells. Okay, so let's read through this really quickly. In non-walled cells, such as animal cells, the movement of water into and out of a cell is affected by the relative solute, the material that is dissolved, concentration on either side of the plasma membrane. As water moves out of the cell shrinks. If water moves into the cell, it swells and may eventually burst. In walled cells, including fungal and plant cells, osmosis is affected not only by the solute concentration, but also by the resistance to water movement in the cell by the cell wall. This resistance is called turgor pressure. The presence of a cell wall prevents the cell from bursting as water enters. However, pressure builds up inside the cell and affects the rate of osmosis. Remember, osmosis is the movement of water. This is throwing it back to when we were talking about movement of water and osmosis in your bio classes. We're going to talk about water potential first, right? Water movement in plants is important in water transport from the roots into the shoots and the leaves. You will likely explore this specialized movement called transpiration in another lab investigation. So we will, but we're going to talk about how water moves and why water moves at a molecular level first and talk about water potential. So in order to understand water potential predicts which way water diffuses through plant tissue. So water moves, we know that. It's a molecule, it's free floating, and diffusion is just the natural flow of flowing from high concentration to low concentration. And when we talk about water, right, some substances have more water in them and some substances have less water in them. And we abbreviate it in a formula as the Greek letter psi. So when you see this kind of trident fork shape, that's going to be talking about water potential. So water potential is the free energy per mole of water, and it's calculated from two major components. It's the solute potential, which is psi with an S for solute, and that's dependent on how much solute you have, right? The concentration. If there's a lot of salt or if there's a little salt, if there's a lot of sugar or if there's a little sugar dissolved. And then the second one is the pressure potential, which is psi with a P at the bottom, which results from the exertion of pressure, either positive or negative, on a solution. So if you can put something in a vacuum and put complete pressure or remove all pressure from the surface of a container, or if your pressure container is closed, there's a different level of pressure inside of it. We're not actually really going to worry about pressure potential just because our containers are mostly open containers that we're working with. So that being said, it means that our pressure potential is always going to be 0. So we're only really ever calculating for solute potential. And I'm just going to say that now because it makes your calculations a lot easier and it's something that you don't have to worry about. So the solute potential is also called the osmotic potential. And this is the formula that we use that was explained up here, right? It says that water potential is pressure potential plus solute potential. And if pressure potential is always 0, then in our calculations, water potential always equals solute potential. Okay, so water moves from an area of high water potential or higher free energy to an area of lower water potential or lower free energy. Water potential measures the tendency of water to diffuse from one compartment to another compartment. So for example, if you have 2 containers of different kinds of solutions and you pour them into one big container that has a semi-permeable barrier in the middle, which means that some things can move through it, but not all things. So in this barrier, for example, NaCl cannot move, salt cannot move because this barrier doesn't allow ions to freely move. But this semi-permeable barrier does allow water to move. It is polar, but it is tiny. So if you have pure water on one side, 100% H2O, and you have 2% NaCl, which means 98% H2O, then there's going to be higher water potential just because there is more water on this side. And so because there is more water and a higher concentration of water, we know that water will diffuse from the higher to the lower, and water molecules will move slowly from the left side to the right side. And as it moves slowly from the left side to the right side, the water level here is going to start to go down, and the water level over here is going to start to go up. And you'll actually see a level difference that will be created because the water potential causes water to move from a high concentration or a high pressure to a low concentration or low pressure. And this is the formula that we're going to be using. This whole page is basically explaining that water potential for when we calculate with using solute potential is based on a negative I CRT. And the I is the ionization constant. So for example, when we talk about ionizing, right, we're talking about things like salts breaking down. For example, NaCl, when you put it in water, it breaks down separately into Na and Cl. So that means that inside of water, NaCl or table salt is actually not one piece at a time. It's not one molecule of NaCl at a time. It actually breaks down into two pieces at a time, which is Na and Cl. So for example, our ionization constant of NaCl would be 2. But you can have something like sugar that you put into water as well. Sugar, or like glucose, for example, does not actually ionize because it is a covalently bonded molecule. Okay, so sugar does not ionize, and therefore it doesn't break apart into multiple pieces, and it stays as one big glucose molecule at a time inside of water. So your sugar constant then, so your ionization constant for sugar then would be 1, because there's only one piece of sugar at a time. C is the molar concentration, and that's usually given in the problem. Okay, it's the concentration in molarity, and I'll explain that in a second. R is the pressure constant, and this is what we will always use. R is always this constant. R equals 0.0831 liter bars per mole Kelvin. And you don't really have to worry about these units because if you take AP chemistry, you will. But you don't really have to worry about these units because they're just going to be canceled out and you'll be left with bars at the end as your solute potential. And T is the temperature, but we want to use temperature in K or Kelvin. We are not going to use Fahrenheit and we are not going to use Celsius. But luckily for us, the Kelvin scale matches up with the Celsius scale. But we actually use numbers that are relevant and in like not too big or not too small. So we would just be adding the base number for Kelvin for 273 plus the current room temperature at Celsius that we are using during the experiment when we're moving water around. Okay, so as an example, a 0.15 molar, capital M stands for molar solution of sucrose at atmospheric pressure. Since it's open to the atmosphere, our pressure potential, remember, is going to be 0. So we don't have to worry about that. So we're only going to be calculating solute potential. And 25 degrees Celsius has an osmotic pressure potential of negative 3.7 bars and a water potential of negative 3.7 bars. A bar is a metric unit of pressure. So our results for whenever we calculate psi s is just going to be bars. So that is our unit. And is the same as 1 atmosphere at sea level. So a 0.15 molar NaCl solution contains 2 ions, which is Na and Cl. Therefore, I equals 2 and the water potential is negative 7.4 bars. So when you compare this, actually, the concentrations are both 0.15, right? So they are the same. But because NaCl breaks down into Na and Cl as 2 separate ions, the I for this solution is actually 2, whereas sucrose does not break down. So the I for sucrose is 1. So basically, these two formulas are calculating the exact same things, except when you get negative 3.7 and negative 7.4, the only difference is because NaCl is breaking down into 2 things. So when a cell's cytoplasm is separated from pure water by selectively permeable membranes, water moves from the surrounding area where the water potential is higher into the cell, where the water potential is lower. Because of solutes in the cytoplasm, okay, so concentrations matter. And the thing that is dissolved as a solute also matters. That's why we talked about the ionization constant. It is assumed that the solute is not diffusing. Okay, in this membrane, we're going to say that the salt or the sugar is never moving. We'll only say that the water is moving. The movement of water into the cell causes the cell to swell, and the cell membrane pushes against the cell wall to produce an increase in pressure. This pressure, which counteracts the diffusion of water into the cell, is called turgor pressure. So if you look at a potato cell here, and there's not that much, so this is pure water on the outside. So obviously we already know water is going to flow into the cell because water never flows towards pure water. And so the potato cell has a negative 3 as its solute potential, which means its water potential is negative 3. It's 0 and -3 compared, -3 is smaller, which means that water is going to flow from the higher number to the lower number. So water is going to flow from the solution into the potato cell, and it's going to flow and flow and flow and flow until it fills up the potato cell. But the potato cell is a plant cell, so it has a cell wall, and it won't flow until it bursts. However, the difference is kind of small from 0 to -3. It's only 3 in difference. So it'll fill until the potato cell reaches a 0 as well. So once these two water potentials are equal, then water stops diffusing unevenly, and then it moves back and forth between the cell and the outside solution at an equal rate. If solute is added to the water surrounding the plant cell, the water potential of the solution around the cell decreases. If enough solute is added, the water potential outside the cell is equal to the water potential inside the cell, and there will be no net movement of water. However, the solute concentrations inside and outside of the cell are not equal. Because the water potential inside the cell results from the combination of both turgor pressure and solute pressure. Okay, so here we have an example of where solute and water is added. And now the difference between the two has changed. Okay, they're basically going to have water molecules move either out of the plant cell or into the plant cell until these two potentials are equal. Once they are equal, then it's balanced, and then water moves in and out at an equal rate to maintain that equilibrium. So if more solute is added to the water surrounding a cell, this is the calculation from before. We don't actually have to worry about this kind of example, so I'm just going to move on from it. But this is another breakdown of the formula that we'll be using. So again, water potential, which is psi equals pressure potential plus solute potential. And we usually don't have to worry about pressure potential because it equals 0 when the container is open to the atmosphere at atmospheric pressure. So that means whenever we calculate these, we'll basically only be using solute potential, which is calculated using -I CRT. Again, I is the ionization constant. Be wary of salts. Things that are not salts usually have an I of 1, but salts can have different numbers based on how many pieces they break apart into once they touch water. The molar concentration is just the concentration of a solution, and you can usually find them based on a capital M. Pressure constant is always this with its units, so make sure you include the units. The temperature is going to be the room temperature that this setup is at in Celsius plus 273. This is literally what I just explained, so if you want to pause and read through it, that is fine. So let me do an example for y'all, and then you're going to do your own worksheet. So calculate the solute potential of a 0.1 molar NaCl solution at 25 degrees Celsius if the concentration of NaCl inside the plant cell is 0.15 molar. Which way will the water diffuse if the cell is placed into the 0.1 molar NaCl solutions? So basically you have a plant cell and you have a solution, and you're going to put that plant cell into the solution. And then something is going to happen to the cell. Either water is going to go in or water is going to go out. And we have to figure that out based on using solute potential. So first of all, we're going to calculate solute potential of the solution. So we know that we're going to use this formula right here. Solute potential equals -I CRT. And we're going to sub in our numbers. So our I is going to be 2 because remember NaCl breaks into Na plus and Cl minus. So there are 2 pieces when it breaks down. The C, or concentration, is 0.1 molar because that's given to us as the concentration of how salty this solution is. Our R is a variable that is a constant that is always 0.0831 liters bar divided by mole Kelvin. And then we have our temperature in Kelvin, which is going to be 25 degrees Celsius plus 273 as the base, so that the unit overall here is Kelvin. And once you calculate that out and you cancel out these units, right? K cancels out with the K on the bottom here. Molarity is moles per liter. So the mole on top here cancels out the mole on the bottom. Liters on the bottom cancels out the liters on the top. So the unit we're left behind with is bar, which is a pressure, which is good, which means we're on track. So our solute potential in our solution that we're going to be dropping the cell into is -4.95 bars. We're also going to calculate the solute potential of the 0.15 molar NaCl plant cell. So this plant cell, it has a little bit of saltiness in it as well. And we're going to calculate out what its solute potential will be. So again, same formula. Let's sub in the numbers. The ionization constant is still going to be 2 because we're still using NaCl, which breaks down into Na plus and Cl minus. The concentration here is a little different because it is 0.15 molar, which means this plant cell is salty, more salty than the solution. The R constant is still 0.0831 liter bar per mole Kelvin, and then our temperature is the same as above because we are doing this experiment in the same room with the same temperature. And so once you sub in the numbers, you get negative 7.43 bar. Now, be careful. When you're comparing these two, remember they are both negative numbers. And larger numbers that are negative are technically smaller numbers. Okay, the magnitude of this number is bigger, right? Because it's a 7. But because it's on the negative scale, it is further away from 0, so it is smaller. Okay, so you're going to take and compare the two solute potentials that you got. Remember, the water potential for both is 0, so we don't have to worry about that. So solute potential equals water potential. So our conclusion here is the solute potential of the 0.15 molar NaCl plant is lower, right? -7 is smaller than -4. Then the 0.1 molar NaCl solution with -7.43 bar of the plant cell compared to -4.95 bar of the solution. Therefore, water will diffuse from the solution into the plant cell because water will always flow from the higher potential to the lower potential. And this is how you make your conclusion based on calculating these two. Okay, so this is in your worksheet in the following assignment, and you're going to be doing basically in 1, you're going to calculate a solute potential for a solution. In 2, you're going to calculate solute potential for a separate solution. And then in 3, you're going to tell me if these are connected together with a semi-permeable barrier, which way will the water move? Will it move from the sucrose to the NaCl, or move from the NaCl to the sucrose solution in number 3? You also have 4, 5, 6, which is the same idea, 1 solution, 2 solutions. And number 6, if you put these 2 solutions next to each other with a semi-permeable membrane, which way will the water move? To the KCl or to the glucose? This is kind of the picture of the setup I meant. So you're going to take these 2 solutions and pour them into one side of a barrier and then the other side of the barrier, and you're going to tell me which direction it moves. We're also going to calculate molarity here. And so this is a separate assignment. It's also not very long. But you want to use your periodic table that I provided a long time ago, and I will reattach in this link. I think it's a good idea for you to save as a document because you will have to refer to it multiple times. But you're going to record the mass of hydrogen, and that comes from your periodic table. So you basically go into your periodic table and look at the atomic mass units of hydrogen. And over here, you can see that it's 1.0. We'll do 1.01 because we're going to round it to the hundredths. And so you have 1.01. And let's use the units of grams per mole. We'll do it for oxygen as well. Okay, so you want to go find oxygen on your table, and we see that it is 15.9994. And if we round that, that actually is just going to be 16. Okay, so this is going to be 16 grams per mole. And I want you to do that for the rest of these. Okay, so you can pause and check, or you can just do it with me, but it's better if you get used to doing it on your own. So if you look at carbon, it's 12.01 grams per mole. And then for chlorine, we can see over here that is 35.45 grams per mole. And that sodium over here is 22.989, so 22.99 grams per mole. And you got to get used to navigating the periodic table, even though this isn't chemistry, but it's still good info to know. So now we're going to calculate the molecular weights of these molecules. And I'm going to do one for y'all, but you all are going to do the other ones by yourself. So we're going to do glucose. And I gave you the chemical formulas because that is the most important part right now. So you have all of the actual chemicals listed up here. Notice that there are subscripts here that tell you exactly how many times you're going to be using these numbers. So one glucose molecule here. Let's do one with the H2O and one without the H2O. So we'll do one here with the H2O and without the H2O. Okie dokie. So here we have C6, which means you're going to be doing 6 times the carbon weight, which is 12.01 grams per mole, plus 12 H's. You need 12 times 1.01 grams per mole plus 6 O's, which is 6 times 16 grams per mole. And you're going to calculate that out on your calculators. Let me do that real quick. 6 times 16 plus 12.12 plus 6 times 12.01. And so our molar mass of our glucose is going to be 180.18 grams per mole. And that's just the unit that we're using. Okay. You can also do the same exact thing and add it with water, because when you put glucose into water, it automatically combines with water. And so the molecules are actually glued together almost. If you want to find out your glucose weight, which you already know, and then you're going to add on the water molecule weight. So you're going to add on, because there's 2 hydrogens per water molecule, and then you're also going to add on 1 oxygen weight, because you know that there is 1 oxygen per water molecule. And so this combined kind of element of glucose plus water, so when you put it into water, is going to be 198.2 grams per mole. I'm going to ask that you do the rest of these two. So you still have to do table salt, table sugar, which is sucrose. It's what you have at your dinner table. And you also have table salt, which is the salt that we consume on a daily basis that you put in your food. And I'm going to grade those for y'all. Now we're going to actually calculate molarity. And molarity is not that hard to calculate because it's how do I explain this? The molarity is always in 1 liter of solution, how much solute did you put in? Okay, so we're basically doing the amount of solute per 1 liter of solution. So the reason we do this is because 1 liter is a convenient amount of solution that we're going to make, and it's easy to calculate using the molar mass. So basically they're saying you want to make 1 molar solution of each molecule. And we know that for glucose, for example, we already know that the molar mass is 180 was it 12? 180.18, my bad. So we know that it's 180.18 grams per mole. Okay. You want 1 mole because you want a 1 molar solution. You want a 1 molar solution. So you're going to want 1 mole, right? If you get 180 grams, 180.18 grams of glucose, then you're going to have exactly 1 mole. So you're going to put 100, let me type in here, 180.18 grams of glucose is 1 mole. So you take 180.18 grams of glucose and add it to 1 liter of water. That will be your solution of 1 molar, 1 molar or 1 molarity solution of glucose. Okay. This is kind of a simple calculation though, because it is in, it's only asking for 1 molar. So there is a more complicated version of this. Let me do an example. So if you want a 2 molar solution of glucose, it's a little bit different because now you want double the concentration. So remember, 180.18 grams of glucose is 1 mole. but you want 2 molar. So you're going to do 180.18 times 2, which equals 180.18 times 2, which equals 360.36 grams of glucose, and add it to 1 liter of water. And that will give you a 2 molar solution of glucose. So your amount that you're adding is based on the molecular weight we found on the other page for sucrose and for NaCl. But then your amount that you're going to be taking of that is going to be based on the actual concentration they're asking for, right? 2 molar means that you're multiplying by 2. 1 molar means that you're multiplying by 1, which is why this is just the original number. So you're going to finish your calculations on molarity. Okay, because we're going to be doing an osmosis and diffusion lab coming up. So let me go over it really, really quickly, and then you don't need to take notes on this. I just want you to go over it, okay? Questions that you can consider as you move into this lab is, when you have an IV at a hospital, why does it need to have salt in it, right? What happens if you're giving pure water in an IV? You want to think about water potential, right? Pure water potential means that water is always going to go into your cells. Is it good to always have water going into your cells? Is that the point of an IV? And how would you determine the best concentration of solutes to give a patient in need of fluids before you introduce the fluids to the patient's body? Because if you give them pure water in an IV, you can kill a patient, okay? So we're going to be using dialysis tubing in our lab on Wednesday, and we'll be using separate solutions, either pure water, sucrose at 1 molar, salt at 1 molar, or starch at 1 molar. And I'm going to be creating these for you, but based on the molarity calculations that we just did, you should be able to know how to make these. And then we're using cups and balances, and we're going to be weighing and looking at color observations of the dialysis tubes with contents based on these. And so we're going to be setting up like this. For example, we'll have an inside solution, which is going to be one of these 4, an outside solution, which is going to be another one of these 4, and then we're going to have dialysis tubing that's holding them separate, and dialysis tubing acts like a cell membrane. Okay, so we're going to be doing 2 separate ones as a comparison to each other. And if you have starch and you have iodine on the outside and the iodine diffuses in, you'll be able to see the starch turn black. If you see the starch go out of the membrane, then you'll actually see the outside turn black. So we're going to be trying to observe for color changes, and we're going to be observing for water changes, because if water is moving into or out of the cell based on what's dissolved as a solute on the inside or the outside of the cell, this weight will change. So we're going to weigh it before and weigh after to see did it increase in weight. Okay, so for example, we'll probably do one that has like water, pure water on the outside with iodine, just to see if starch moves in or out. And we'll do the starch solution on the inside, again, to see if starch moves in and out. And then we'll be, sorry, that was the color test. And then we're going to be doing a massing test to see if, based on the starch or the pure water, which way the water is moving. If this increases in mass, then that means water is moving in. If it decreases in mass, that means water is moving out. So it's a pretty interesting experiment that observes both diffusion and osmosis. And then for Wednesday, I want you to think about this beforehand just to be prepared, right? So write an if-then statement predicting what will happen to the movement of solutes. So if starch diffuses across the semi-permeable membrane, then the outside of the beaker will turn blank. Now, I want you to also make a hypothesis. If the water diffuses across the semi-permeable membrane, do you think it's going to go into or out of the tubing or the cell? Because if it goes in, then the tubing will start weighing more. And if it goes out, then the tubing will start weighing less. So you can make predictions based on that at the start of the lab on Wednesday. This is the basic kind of, setup that we would be doing in person, but I will be doing for you during the demo. And then we'll be writing conclusions based on the amounts moving in and out based on mass. And we'll be doing a color conclusion based on the reaction of starch with the iodine, if there is any. Again, make sure that you complete your water potential calculations due today and your molarity calculations. If you have any questions, make sure you ask during class, or you can ask through Remind or email. And then your Chapter 5 Summary and Learn Smart are due Friday, as well as your cell drawings based on the textbook pages.